Karen, for accepting our invitation for this Astrobiology Webinar. It's quite important for the students in India. Dr. Karen, she's from the Open University. She's now the Director of Astrobiology at the Open University. And she does a lot of work with the free analogues on planet Earth. I think it's important that we hear from Dr. Karen. So Dr. Karen.
Thank you.
Yeah, thank you.
Thank you. Thank you for that introduction. As we said, my name is Dr. Karen Olsen-Francis. I'm Director of Astrobiology OU, which is based at the Open University in the United Kingdom. So I'm going to use this opportunity to talk about some of the work that my team is doing here in the UK. I'm going to focus on the work we're doing around Analogue and lab simulation experiments. We do a lot of work looking at icy moons and Mars, but for this talk, I will focus on Mars. So, are we alone in the universe is one of the most compelling questions of our time and is the driving force behind the field of astrobiology. And to address such a question requires a multidisciplinary approach. So this is where we need to bring different scientific disciplines together. So, for example, we have microbiologists like myself, organic geochemists, physicists, geologists, working together to address these questions. And looking for evidence of life elsewhere in the solar system and beyond has been the driving force for the robotic space exploration. Over 50 years, we've had this dream of going to space and finding evidence of life. And then we look forward over the next 10 years, we have a number of missions planned. These include the icy moon and Mars. We have Mars 2020, which will be going later on this year. We also have the European and the ESA ExoMars mission planned for the next couple of years. And Mars 2020 is the beginning of the first of the Mars sample return mission, which eventually we will get samples back from Mars to Earth, where we can look for evidence of life and also trying to understand if life had evolved on Mars. So for this talk, I'm going to give you an overview of life from a microbiology perspective. Then I'm going to talk about some of the work that we can do to study life on Earth using both analog experiments or field work and laboratory simulation experiments, before going on to talk about some of the work specifically Astrobiology OU is doing, which is kind of over the last couple of years, we've evolved past the traditional science boundaries into the social science areas. We've received an investment of nearly £7 million by the UK government, and we started to look at the governance and ethical commercialization impact of astrobiology and the effect that this can have. And then I'm going to finish the talk by looking just highlighting some potential collaborations that and funding opportunities that are available for Indian citizens to come to the UK to potentially work with us. So I thought that would be an opportunity for you to think about, the next step in your career. So what do we know about life? So when I think about life, I'm focusing on life as we know it on Earth. And what we're focusing on is microbial life. So this is very, simple life, unicellular life. And for life to exist, and there are some key requirements. The presence of liquid water is essential to when, where, and what conditions life could have evolved or exist on present day or early conditions in the solar system. And without liquid water, cellular function would not be able to occur. The cells wouldn't be able to get rid of byproducts or toxins, and they wouldn't be able to take up nutrients. But life doesn't just need liquid water. It also needs key elements, the building blocks for life. So for this, we need elements such as carbon, nitrogen, oxygen, hydrogen, and phosphorus. And these make up the building blocks for life. Phosphate is particularly important because it's part of the key molecule ATP, which is used as the currency within the cell for cellular activity. And we also need trace elements and major elements. So, for example, iron is a key component in enzymatic reactions. And for cells to exist, they need to have a form of energy. We eat, for example, we take a chocolate bar and we eat that and it gives us energy. On Earth, primary production on the surface of Earth is driven by photosynthesis. So we take energy, plants take energy from the sun to produce energy from the sun for their function. In the subsurface of Earth, and probably more applicable for when we look beyond the Earth, is chemolithotrophic metabolism. And this is where organisms take energy from inorganic substrates, for example, hydrogen, sulfate, and iron. But even if we have all these key ingredients for life, we also need to make sure that the physical chemical parameters are conducive for life. And this includes things like pH, temperature, water activity, and pressure. And so all these conditions need to be right for life to exist and for an environment to be habitable. And when we look at Earth, life can be found nearly everywhere. Some of the, microbes, particularly those that are called extremophiles, are microbes that adapted to live in some of the most extreme environments on Earth. Some of the first examples of these are thermophilic microorganisms. These are microorganisms that live at high temperature. Over 30 years ago, some of these organisms were identified in Yellowstone, in the hydrothermal vents there. And since there, our understanding of extremophilic microorganisms have evolved. We now know examples of microbes that live in extreme pH, at high pH, so alkaphiles that are above pH 10, to low pH, so those, for example, below pH 3, and right through to microbes that can survive and potentially live within salt and brines and salt crystals on Earth. So we use this information of what we have on Earth and looking particularly at these extreme environments to look at where life could live and what environments are habitable beyond the Earth. Now, as I mentioned previously, I'm going to focus this talk on Mars because it's a hot topic with Mars 2020 going in the next couple of months and the Mars sample return missions planned. And when I think of Mars as a microbiologist, I kind of look at it in two stages of habitability. There's the early day Mars and present day Mars. Because today, if you looked at the surface of Mars, there's not evidence of large surface water. Microbial life on the surface would be exposed to high radiation, desiccation conditions, oxidation. It'd be really detrimental to life. However, on early Mars, the conditions would have been more conducive to life, we believe. We think the climate was warmer and wetter, and that surface water could exist. And while these conditions on early Mars are also similar to those on early Earth, and potentially as life evolved on early Earth, as we know, life could have evolved in a similar manner to that on Mars. Or even more, if life hadn't evolved on Mars, it could have been transferred from Earth or any other planet through lithopanspermia, which is a process where life is transferred from one environment to another. So an example of early Earth environment which potentially could have been habitable is a lot of information regarding this has been identified from the Mars Science Laboratory mission to Mars, which is focused on Gale Crater. As you see in this picture here in the top left, this is what some of the images that came back from MSL demonstrated: a very dry, arid environment with some evaporites on the surface. Whereas on early Mars, this condition here is an image which is these large fluvial systems. The paper here on the right is a pivotal paper in understanding of habitability. And this is where it demonstrated that the MSL science team suggested that the water in this Gale crater, this was a large fluvial system, and the composition of that brine water could potentially have supported life. For example, the key elements that I mentioned previously, such as carbon, hydrogen, nitrogen, and sulfate, have been thought to have existed within these environments. And also evidence suggests that there could have been redox couples available for chemolithotrophic metabolism, which would allow the microorganisms in these environments to grow and function. Although this is condition, so Gale Crater is predicted to have had this kind of large surface water area during the Hesperian period of early Mars. But as the environment on Mars evolved to what we have today, the water on the surface water evaporated with the atmosphere as the atmosphere evolved, and the conditions today on the surface of Mars are very different to those on early Mars. So the conditions today are very dry and arid, as I mentioned. And there are lots of evaporitic deposits on the surface of Mars. So, for example, there's these crystal structures similar to this, what we have on Earth. And if we're talking about habitability from a present day Mars perspective, we can look at this in a different environments again. So the surface of Mars, within these sulfate evaporite deposits, which we could potentially have had the last remnants of life from these large early fluvial systems which were predicted to existed on early Mars. And on Earth, we know that these salt crystals can actually be refuge for microbial life and biosignatures. And we know that on Earth, microbes can stay encapsulated within salt crystals and survive for thousands of years. We can also think about it in the subsurface environment. So in the subsurface environment, liquid water could potentially existed, and it also would be protected by the detrimental effects at the surface. An example of playing to this is the ExoMars rover, which has a 2-meter drill on it, with the idea that within the 2-meter zone, any life within there would be protected from the detrimental effects of the surface. Now, in this near subsurface environment, the water would be very salty in composition due to the conditions at the surface, but in the deep subsurface, liquid water could exist. And again, this is somewhere that microbial life could form and exist. So to understand, these missions that we have going to Mars, it is plausible that Mars was or is habitable. And to look at this in more detail, we have a number of lag detection missions going in the next couple of years. And for these missions, what they need to do is to identify unique biosignatures which could be used to demonstrate that microbial life has or is still existent on Mars. And there's a number of examples of what could be used for this. One is organic materials. So these are organic molecules that make up cells, which could potentially be found on Mars. Or if we're looking at biosignature that can persist over long timescales, we're looking at geological biosignatures. So this is where microorganisms can affect the local environment to affect the production of secondary alteration minerals. An example of this is sulfate-reducing bacteria, which play an important role in mediating the formation of certain sulfate minerals. And some of you may have heard the big fuss over the last few years about methane on Mars. Now, on Earth, methane is a biosignature for life. And on Earth, over 90% of methane is produced by biological activities. So measuring how methane, particularly, for example, methane to ethane ratio, is something that the TGO orbiter is doing around Mars at the moment, is being used as potential as a biosignature for life. So seeing if fluxes in methane can be, is being produced by biotic activity. And to study habitability and biosignature formation, we need to kind of look at microbes on Earth to have an understanding before we go to these planets to look for evidence of life. One way of doing that is to look at microbes under simulated conditions. So for this, we can use simulation conditions on Earth. Here's an image of our Mars environmental chamber that we have here at the OU. And what you can do is you can put in a Mars gas, which is predominantly comprised of CO2. We can simulate solar radiation using a xenon light force, and we can do temperature fluxes and freeze thawing using liquid nitrogen. And again, a lot of the work we do is focused on the surface of Mars with this chamber, and we can change the pressure within the chamber to simulate the conditions on present day Mars as well. But if we really want to simulate the radiation that we would have had on the surface of Mars, we kind of need to look further afield. So one way of doing that is experiments in low Earth orbit. So here in this picture here, we see examples of Biopan, which was a European exposure experiment which had microbes, in samples of microbes circulate the Earth for 10 days, returning to Earth so we could look at how microbes survive. But for long-term experiments, we've been using the International Space Station to do this. There's a huge European consortium called through Biosign and Biomex, which have been looking at how low Earth orbit and simulated Mars conditions can affect both survivability and biosignature formation under these conditions. So I've been involved in working in these. One of the work that I was doing was looking at if we could isolate microorganisms from Earth that could survive these conditions, which could then be used to kind of infer what we would look for biosignatures on Mars. So for part of this work, what I did was we took samples from the coast in the UK from a place called Bear, and we took this. It was a community of cyanobacteria, and we exposed them onto the International Space Station for nearly 2 years, nearly a year and a half. Sorry. What we did when we got the samples back was that we were able to isolate microorganisms that were able to survive these conditions. So here are some images that we had. So what we found was that none of the this was a Gliocapsa species, and as you can see, it's got this large extracellular polysaccharide material surrounding the cell, which offer protection. And also what we found was they formed these massive colonies. And what we predicted was that these kind of colony formations were able to help protect and shield some of the microbes within the colony from these detrimental effects. Now, we weren't able to isolate microbes from full exposure, but when we looked at, about the 200 filters, using some of the filters, we were able to isolate this microbe. And what we're able to do also then is to look at how biosignatures would be affected by these conditions. So here's an example of some Raman spectra. And as you can see, as with a different exposure, we started to lose signal. So this kind of information on the ISS, when using these ISS experiments, we can start to understand, firstly, what are the requirements for life to survive under these extreme conditions? And secondly, what organic biosignatures could be used to detect them? But, not everybody can do these simulation experiments. And I think it's also really important to look at some of the work that you can do in the field on Earth. So looking at Earth to understand, and using that information to understand what could exist on these potentially habitable environments. So we do a number of field studies where we go into the field and look at analog sites. So analog sites are sites which are similar to the conditions that you want to study. And depending on what environment, and particularly Mars, what time in its history you want to study, you can then identify a field site which could be used to study that in more depth. So here is an example of some of the field sites that we've been to over the last year. I'm going to talk about Lost Hammer in a minute. Last year we did some work in Dalal, which has been deemed one of the most extreme environments on Earth in Ethiopia. It has a pH of about 0, high temperatures above 100 degrees, and it's really saline and toxic. Even with working conditions here, you're looking at, you have to go up really early, and the conditions, you're looking at 30 degrees above. I think the maximum temperature that we had at the camp where we were staying was 50 degrees. So it's a really hot environment, but we were able to show that microbials could survive in this environment, although they may not be able to live, but there's evidence of life we could detect in these environments. We've also recently been working over here in India. We've collected some samples for the Rana Kutch, and I think you're very lucky living here in India because there's a lot of analog sites on your doorstep. You have the lunar crater, which is a fantastic analog, and particularly at the moment, now it's with the changing of the water to pink, which seems to suggest that it's become very evaporitic. These kind of analog environments that you have on your doorstep could be used to study potentially processes that could have existed on Mars. We've also been recently this year over to Botswana, where we've been working on some paleolates. Again, these are highly salt environments, and they're used to look at survivability of microbes and what microbes can live within these highly salt environments. We've been doing some cores here as well, so we can get a transaction of how microbial communities change with salinity and depth. So depending on what period of Mars or what environment that you want to study depends on what you can use for an analog. So as I said, looking at present day Mars compared to early day Mars, you're looking at very different environments. So some of the work that we've been doing is we've been looking at sulfurous and saline water, which is proposed to have existed on the surface of Mars during the Nicaean and early Hispanian period. And we know there's lots of sulfates on Mars, and we're predicting that the environment, the liquid water in this environment would have been very sulfate rich. So for this work, we've been working in the Axel Heiberg island in high Arctic. This paper actually just got accepted this week. And basically, this is an environment which is characterized by polar deserts, and it's exposed to it's a continuous permafrost environment, and it hosts a series of sulfur-rich cold springs. So we've been particularly focusing on Colour Peak and Ross Hammer. There's been a few studies over the years, but not looking in depth at the microbial community, particularly at the Peak, Ross Hammer. Sorry. So what's so special about these springs? Well, they're really rich in saline and sulfate, so they're very much really unique in that perspective. And what we were using this was as an analog for Mars during the late Noachian period. We came to the conclusion that this was a great analog for us to use. What we did was that we did some modelling where we took brine compositions of what we know that have been used to predict what the water composition was on early Mars, based on the geology, and we mapped out, we compared that to the composition of Colour Peak. So as you can see here, the sample of the water that we were testing were very similar to some of the sulfur vein fluids which would have been predicted at Gale Crater. So as a microbiologist, I was very excited to use this to look at the microbial communities within these sites, and to use that as kind of to understand what potential microbial processes would have existed during this period. So for this, we used a combination of both 16S analysis focusing on DNA, but also cDNA, so we could look at the active community within these brines, not just the dormant community. And what we found from this analysis, so what we did is we collected water and sediment samples, and the results here we're showing are the sediment samples. So this is kind of the anoxic zone in the sediment. And what we showed was that they were dominated by gamma proteobacteria, particularly that of the Bacillus, which you can see here, which is demonstrated by the purple in the bar chart. And what was interesting about this, and kind of what we were able to predict, that this is a diversity with sulfur-related metabolisms. So this includes sulfur oxidation, so primary reduction, we thought based on this, would be driven by sulfur oxidation reactions. And there was also a lot of sulfate, so we're expecting there'll be a lot of sulfate reduction going on in these environments. So very much dependent on the sulfur cycling in this environment. So based on what we did, what we knew from when we compared this to the water conditions that we predicted for the early Mars, we're interested to understand how, could microbial life on Mars be driven by sulfur cycling? So what we did was we used thermodynamical feasibility by calculating the Gibbs free energy reaction for some of these pathways. So what we did was that we used the brine compositions that have been determined for this period, and we included elements based on what we know about the existence of Mars, looking at oxygen levels and nitrate. What we did is we calculated the feasibility of sulfur cycling on Mars based on the Gibbs free energy calculations. And what we found was that based on what we predict, the conditions that we predicted existed in this period, we found that aerobic sulfide oxidation could potentially be dominant primary production in this environment, but also it could be fueled by denutrification, could also be fueled by sulfide oxidation. So the idea would be that primary production in this environment could have been driven by having the sulfur oxidation, but as these microbial communities have grown, they would have secreted carbon and their cells would have died, which would have led to heterotrophic microorganisms existing. So these are microbes which basically would have eaten the primary producers to kind of form a community. So what we predict in these environments on early Mars, that it would have been very much sulfur-driven environment, ecosystem, biosystem going on, where primary production was dependent on sulfate, and this led to heterotrophic metabolism occurring, which could utilize the carbon and the organics that are produced by these microbes. But not all environments that we need to look for analog sites need to be that extreme. Going to the high Arctic, it's very expensive, it's very difficult to work in, and you only have a one shot at that. So for some of the work that we went on to do with simulation work, which I'll talk about later, we wanted to find something a little closer to home. So what we did was we shifted our focus within in the Mars history to look more at the early Hesperian time, where Gale craters are thought to have existed. This was a large fluvial system, and what we knew about the conditions at Gale crater was that the temperature fluctuated between freezing and just above. The pH was circa neutral. Potentially, as the water started to evaporate, we'd have a water. So, the composition would evolve from 1 to 2% right up to saturation. We knew that there was a varied. The paper that came out about Yellowknife suggested that there was a varied redox state. And we thought that as the lake started to evaporate, communities would have been exposed to evaporation. So what we did was we did some work a few years ago where we wanted to find an environment close to home which mimicked these conditions, and that we could then use the microorganisms to look to do some of the work that we want to do in our simulation work. So for this, what we did was that we focused on the intertidal zone in a place not far from where I grew up. And what we found was that we found this community in the below the redox potential zone. So it's a very anaerobic environment. Microbes could survive the conditions which we predicted could have existed at Gale crater. And what we found was that the community was really diverse. There was bacteria and archaea in there. There was chemolithotrophic microbes, fermentative, and heterotrophic. So there's this whole selection of microbial metabolisms which could then be used, that could survive these conditions. And we kind of decided that this was kind of like our pet Mars community for the lab. So we've been using this as our kind of starting community for a lot of the work that we've been going on to do. So the work that we're going on to do is lab simulation experiments. Field work is fantastic. It's a great opportunity to go out in nature and see how natural systems would exist. However, the conditions are limited. Field work is limited. You're never going to get a condition where it's 100% similar to what you have in on Mars. And also there's no controls. If you want to look at biosignature production, you're having to look at, you don't have your negative controls, your abiotic controls to compare it to. So what we've been doing at the OU is using lab simulations, experiments to simulate some of these conditions. So yes, no, they're not 100% similar to what the conditions would be on Mars, but they're a lot more similar to some of the analog work that we've been doing. So we have a whole plethora of simulation chambers at the OU. We've got the surface ones, example in this picture and what I showed before. But then we've also got systems where we can simulate flow-through systems, which I'll go on to in a minute. And what we do is that we can then use that to look at biosignature and habitability. So traditionally, when we think of simulation experiments, they're normally kind of done in wheaten bottles like this. So you take some, you make a weak media, which kind of has the ingredients that microbes want to live, a bit of bath salt as an analog block condition, you put in a CO2 headspace, and away you go. But these are very limiting. So what you find is that they're closed systems. So you kind of run out of nutrients really quickly. They're very short, so they only probably run for about 4 weeks, couple of weeks. And also you get kind of like the steady state pH. So as I said, they're very limited. You can also only look basically one stress condition at a time. There's generally, you're using this growth media, so you're kind of helping select the microbial organisms that you want. So we kind of have a bit of a problem. We identified that there was a gap in what needed to be done here. So also, as I said, these experiments are normally run for about 2 weeks. So you're only getting partial dissolution of the rocks. So what we did to address this problem was I started working with a colleague of mine, Suzanne Schwenzer, who's a thermochemical modeler. So she's done a lot of work as part of the MSL team, where she models some of the brine compositions and liquid water compositions that we have used to predict what the composition was on Mars. So the solution that we came up with was a two-pronged approach. One was to use a flow-through reactor, so an open system. This could be used to simulate large fluvial systems on the surface of Mars, but also more related to present-day Mars subsurface conditions as well, because we could alter the pressure. We also linked that also to thermochemical modeling. So this is where we can model things over geological timescales. So the first thing we did was I built this chamber, which is a high pressure chamber, so it can go. So we can use it to simulate subsurface conditions as well. And basically, what it is a reservoir where you can add some brine to it. It flows through a reactor which contains some rocks, some simulant, which I'll talk about in a minute, and then samples are released, and you can kind of continue to sample these. So you kind of have this continuous flow of liquid through the system, which can be used to kind of simulate some of these flow-through systems, which we predict could exist on Mars. So the first thing we did, what we need to do, is to find a Mars simulant to put into the chambers. Now, there's a lot of analogues out there already on the market. GS1 is a prime example. But what we wanted to do, that's a very generic one. So what we wanted to do is kind of look at different environments to start looking at microhabitats. And also what we found was there was huge variation with what we predicted on Mars, what we know from MSL data, and also the composition of these analogues material. And probably most importantly from a microbiology perspective was that they didn't take into account the oxidation state of iron. So as I mentioned before, microbes can use Fe2 and Fe3 for growth, and some of these simulants just didn't take that into consideration. So what Anisha Ramkisson did, one of my postdocs, was she developed a number of simulants which we could use for these experiments. So we developed 4. One is a contemporary global Mars regolith, which we based on the rock nest chemistry. We have a hematite-rich regolith, which is very rich in iron, which is based on the hematite slope. We have a rich sulfur regolith, which is based on Passer-Robles, and we have an early Earth unaltered Mars basaltic shergotite. Now, the basic composition of these were made using various proportions of the following of gabbro and minerals such as dunite, quartz, pyrite, and gypsum. So to make sure that we got the iron correct, what we did was that we used iron silicate glass, which is rich in Fe2, so we could ensure that we had the right amount of iron in this. So what we did was that we were able to develop a number of simulants which had, we were really confident in the results that we got. And what we found was that the percentage difference that we had was less than 5%. So we're really happy with some of the simulants that we made. The composition was less than 5%, difference was less than 5%. So these are kind of what we used as part of our Mars simulants. So here's an example of the hematite-rich composition one that we used, and this was within 4% weight, % volume. And here's a composition of it. And we got the concentration of Fe2 to about 57%. So this is kind of like the basic material of what we wanted to use for the simulations. And based on this composition, what we were able to do then was to develop a brine or the liquid water composition that we wanted to use within our simulations. And for this, what we did is what they use for Mars, and they're doing some of the Mars data, is that we used thermochemical modeling, and this code called ChimXPT, which was previously known as chiller. And so what we do is we put in pure water, and we have a pressure and temperature for one bath, temperature 25 degrees, and we do different water to rock ratios. And what we do is we dissolve the mineral in the models to kind of come up with a composition of a water composition or brine composition, which we could then also put into our chambers. And because there's no nitrate within these brines, what we did was that we used the values that Stern calculated, identified on Mars back in 2015, and we added nitrate air composition of 1.1% weight per volume to these brines. So going back to the chamber, so in the reservoir, we have the brine that we've modelled in there, which is continuously flowing. In the reactor, we have the simulant that we've made. And then what we need to do is, and that's fine for our abiotics. So we always run that first to get our abiotic values to see how that influence, the composition and the secondary alterations that are made from that. So we do the abiotic first, and then we add a biology to it. So the biotic experiments are run then in parallel. But to make our analog community, we do a couple of things in the labs before we get to this stage. There's a few papers out over the last couple of years that we've kind of questioned where they've just gone straight from an analog site into simulation experiments. And we were concerned that there was going to be a lot of crossover organics in these samples, and that would affect the microbial community in this. So what we did was we tested out our theory. So when you go straight from the estrine, you've got a very diverse community. But when you sub it a couple of times in the brines that we developed, what you found that was the diversity of the community decreased over time. And what we did was we used these diluted brine compositions communities in our simulation experiments. So for example, here we take from T7, and that's what we use in our simulation experiments. So they've kind of had time to adapt in the lab, and then we then use them in these experiments. And what we find is when we put them into these chambers, and we can use, we can run these experiments constantly for over 6 weeks, we get a continuous growth rate with a W rate of about 26 hours. So we kind of use that then. We can then use this to look at, long-term effects of microbial growth that can be run for weeks if we wanted to. One thing that we do find over these timescales, which is of interest, is that the pH of these change quite significantly. So the microbes produce organic acids and fermentative material, which decrease the pH of these of the brines, whereas the abiotic can kind of, if anything, increases slightly over the time. And what we do is, we've run these for over a month. That's long term compared to most of the lab simulation experiments that are been run to date. But if we want to look over geological timescales, we link that also to our modelling. So again, we use this ChimXT code used, and this has been used extensively for looking at basalt weathering and applied to Mars compositions. It's also used to look at deep ocean environments. So what we do is we look at, it's a step, so basically we carry out a step-by-step titration of abiotic dilution over different pHs, starting with a high water to rock ratio and then going to a low water to rock ratio. So a low water to rock ratio is where you have a large water surface, so there's increased dissolution. And over time, what happens is that as this dissolves, you get a low water to rock ratio. So you have limited service environment. And the high water to rock ratio is very indicative of water fractions. And what we could do is use this to kind of calculate the effect it would have over geological timescales. So what we do is that we model this at different pHs using the abiotic and biotic system. And what we find is, some prelim data that we can show has demonstrated that when you run this with the biotic and microorganisms in them, what we find is that the secondary alteration minerals that are produced are a lot similar to the abiotic, but a lot more simpler as well. They're not as diverse. So we have simpler precipitation. At a high water to rock ratio, we're getting a lot of goethite, and at low water rock ratio, we have a lot of clays forming. So this is really interesting when we start to think about how we can start mapping this to what the data that we get from some of these missions is, that we can start to understand what secondary alteration minerals could be used for biosignatures. So relating to this work that we've been doing, the conclusion is that a lot of this analog fieldwork and lab simulations, we want to combine that with modelling to kind of understand what biosignatures can be used for life detection. This is just a small aspect of the work we're doing. We're also looking at organic biosignatures. We've just employed a postdoc to do that. And we're also looking at volatile organic acids. So, for example, we're very much looking at things like methane and looking at how they are impacted by the simulation, when we simulate these conditions. We also have a couple of the team looking at icy moon research, so looking at some of the subsurface icy moon conditions. Again, we can use the chambers that I developed to simulate these conditions, so it's a really unique way that we're doing it, but this is something that's feasible. But and this is very much science-based, but over the last couple of years, my group has kind of developed past the science. And I just want to take the next few minutes just to explain some of the other work we're doing. How astrobiology can feed into some social and economical issues and aspects as well. So I've just we've been doing some work over the last couple of years looking at governance. There's planet protection. It's very much about who owns space. When we bring back samples, how do we regulate it? Very much looking at it from a governance perspective. We've also employed people that are looking from an educational perspective. So we're doing a lot of teaching around astrobiology, going into schools, developing teaching material where we can teach STEM using astrobiology as the core. Commercialization, and there's another key area that we're looking at. So using space technology. So for example, one of the members of our team has been using the GC technology that was developed for the Beagle lander to detect TB in some countries as well. So using technologies that have been developed for looking for life from a space context, but actually using it to look for global challenges. And then finally, we've been working to look at global challenges as well. So a project that we've recently been working in is using space technologies to look for areas where would be prone for malaria. So using satellites and drones and rovers to look for areas where surface water could be found where malaria could thrive. And this is something that we've been working with, we've just started working on in South America. And then seeing if we can use microbial spores as a way of deterring the malaria. So this is some of the work going on that. So to do this, I have a team now of 56 people working in astrobiology, kind of falling under these different remits. We've got the education, governance, science, international development, and commercialization. And then if you're interested in learning anything more about some of these key areas, I've given you their names in this slide. And here is also the astrobiology email address. And I'm sure they'd love to hear from you if you have anything specific regarding these areas that you'd like to discuss with them or opportunities. And talking about opportunities, I did mention at the beginning of my slide that I was going to talk about some funding opportunities for you to come over to the UK to study astrobiology if you're interested. So examples of these are the Newton Fund, Global Challenge Research Fund. There's a specific call for the ESRC ICR SSR call, which is for India-UK collaborations particularly. And also there's the Commonwealth Scholarship. So if you're interested in any of these, or if you're interested in using any of these to come over to visit us at the OU, please just email the generic emailer box and we can put you in touch with the right person and help you with your applications. If you're particularly interested in coming over as part of your research to use some of the simulation facilities that we have, the EU-funded Europlanet Research Infrastructure Grant, which was recently funded. If you Google that, you'll find their website. It's possible to apply for funding from the EU to come over to the OU to use our chambers for a couple of weeks at a time. So this would be an opportunity for you to kind of, if you're doing some work and you've got some isolates or something and you want to look at how they survive under Mars conditions, we could do, you could come over to the OU and do that, or run some of the flow through experiments that you want to do. So there's huge opportunity for you there. Or just email me directly and we could discuss this further. So I think that's me. I've made the 45 minutes, which I was asked to keep to. So if anyone has any questions, I'm happy to help. Sorry, I can't hear you, sorry.
Oh, My mic was mute. Okay. Thank you. There are a lot of questions coming from the students. So I'll paste a few of the questions on your WebEx chat screen. Will you be able to see them?
Sorry, where am I?
I just pasted one question on your chat screen.
Okay, let me just grab this up.
On the WebEx. This question is from Divita Gupta.
Yes.
I can read it if you can.
I can see it. Yeah. So there are work being done on fungi as well. So there's a lot of work looking at black fungi in Antarctica because it can survive UV radiation in Antarctica. There's been fungi used in some of the exposure experiments on the International Space Station. There's a group in Italy called Silvano's group in Italy who's done a lot of work in this area. So yes, the fungi have been really are another area that they're looking at. I personally don't work on fungi, but there are groups that are working on them.
Okay, thank you. I'm pasting the next question, but I'll read the question first before your answer. This question is from Suryendra Saha. Can we expect life at the subsurface in the icy moons like Enceladus? Can tardigrada be a potent candidate for microbial life?
So I personally think the icy moons are a really exciting location for looking for habitability, because the conditions in the ice, particularly Enceladus, suggest that maybe they are habitable. But what's exciting about Enceladus is it has this hydrothermic, it's got hydrothermic vents going on, and also it's a closed system. So if there was life in there, it would probably be a second genesis, which would be really exciting. I don't know about tardigrades as a potential candidate, if they would be able to survive in there. But microbial life would be something that could potentially survive and grow in these subsurface oceans. There has been work done on tardigrades on the International Space Station again, and they've been shown to survive. So tardigrades are pretty, hardy stuff.
Mm-hmm. Okay, thank you. There's a question from Dr. Manan Desai. Atmospheric planetary systems are complex systems with multiple variables. How accurately can such systems be simulated? How many variables considered?
Yeah, I think so. For the work we do, we just use a Mars gas composition, which is very generic. I think there's probably groups that are looking at this in more detail, but for what we kind of looking, it's just predominantly a carbon dioxide headspace with minor gases in there as well. So I think you're right. I think there is lots of variation. I think, as I mentioned, lab simulation experiments have their limitation, but they're a good stepping stone for field work to direct to missions, I think.
Mm-hmm. Okay, thank you. And there is one question from Devadatta Das, something similar to the previous one. How the reduced gravity condition on Mars is simulated in lab?
Yeah, so there are special stuff, work that's done looking at gravity, and particularly looking on relating it to for example, on using the International Space Station, relating it gravity to that. The Mars simulation experiments, we lower the pressure. We can simulate the right pressure for the surface of Mars, but we don't really take into the gravity conditions in that. But that's a good question and something to consider.
Okay, thank you. So one more question from Kamarkar. Is abiogenesis still occurring in present-day deep ocean hydrothermal vents? If so, then are there any efforts to study them in situ? I've pasted it in your screen.
Yeah, I think we've kind of evolved from that. I mean, there's a lot of work that's being done on hydrothermal vents at the moment, looking at the microbial communities that exist with them, rather than the biogenesis of them, but looking at maybe the communities, what can exist there. They're very much dominated by chemolithotrophic microbial metabolisms, and looking from an evolutionary perspective. So that there is work done on deep ocean hydrothermic vents, and particularly, interest is becoming a lot more from an astrobiology perspective when you are thinking about, for example, Enceladus, that has these, we believe, have these hydrothermic vents, and also Europa to a certain extent. So there is a lot of interest in these environments.
Okay, thank you. There is one question with respect to olivine being a simulant. Just basically to the screen. Right, I'll read out the question first. Why olivine is used in the brine solution for the simulation experiments? Olivine is usually present in the Mars analog sites, but still, is there any other reason for using that?
Olivine is just, I think we use olivine because we know that it's potentially it's been detected, and also because the Fe2 to Fe3 ratios, it helps with our, determining our ratios. We've done a lot of work looking at iron oxidation with olivine, and it's easy to get hold of. The dissolution rate can support microbial growth. So for us, it's kind of a win-win situation. But there's lots. No simulant is perfect. There's always going to be variation, and everyone has their ideal one depending on what you want. Our analog simulants, are great for growth experiments and biotic weathering experiments. But if you were going to use it, for example, if an engineer wanted to use it to test the ExoMars rover, it probably wouldn't be ideal. So, the simulants have to be taken with a pinch of salt to a certain extent. It has to be dependent on what you want to use them for.
Thank you. And there is one question regarding the sampling from the depths. Just a second, please. Right, so I'll read it first. Are there any estimates on the depth conditions of near surface and deep surface on Mars to be probed for microorganisms or habitable conditions? From Rajesh.
Okay, so the ExoMars rover is going for a 2-meter drill, because it's believed that under a meter you're going to get the detrimental effects of the radiation isn't going to be that difficult. It's going to be, isn't going to be a problem. But then, there could still be life in the near subsurface. It all depends on on the water conditions. There's been a lot of work that's been done at modelling the water composition in the subsurface, so the near subsurface right down to the deep subsurface. So life is dependent on the format, on water being available. So there's no exact depth. It's all dependent on the water. So in the near subsurface, you're going to get really saline environments. So you want to use microbes potentially that are halophilic, so microbes that like salty environments. So the composition is dependent as well. But the drill on ExoMars is going for about 2 meters. So it's kind of variable. There's a lot of work that's been done on that, looking at basically it's linking it to water availability.
Okay,
Would you like me to answer the question before about that Vikram wrote?
Yes, please. Yeah.
So how do we detect presence of any microbial life on Mars and ensure that it's different as it is on Earth? Now, that's a really interesting question. So it kind of links back to what I was saying about Enceladus, is that if we found life on Enceladus, because it's a closed system, we would know that it would be a second genesis for life. But on Mars, it's a very different situation, because yes, life could have evolved on Mars, but also it could have been transferred either firstly by lithopanspermia, or also it could be contamination from that we take there as part of their analytical equipment. So I think this is a very difficult question. I think what we'd have to do is look at look at the biosignatures. So look at the organics using the instruments that we have to get an understanding of what it is, and then kind of defer from there, if it's similar or not. I think the first question anyone's going to ask is, if we find life on Mars, is it from Earth? And I think there's a lot of work that's going to have to go into to prove one way or the other if it's from Earth or if it is actually indicative to Mars.
Oh, thank you. Yeah, life on Mars is a very, very big question. And one question from Andre. Do you think that there scope for identifying new analog sites, particularly in understudied parts of the world?
Yes, and I think we had a quick chat before this, before we started this talk, and I think there's just looking, take India for example, and I know Andre is based in China. There's a lot of under-resourced analog sites in there. We have a lot of the traditional ones that we look at. Lake Tiras in Spain is very much looked at from a sulfur perspective. You've got your hydrothermic vent systems in Yellowstone. But I think, just looking at India, you have so many analog sites here, which are, that you as locals know about that we just wouldn't know about. And I think, look out, look at your back door and see what you have. And, if you identify anywhere that you think could be interested, contact, there's lots of people around that can help you to think about it. As a potential analog. So yes, I think there's a lot of underexplored analogs, and I think as our understanding of as our number of the missions about icy moons and Mars increase, and our understanding increases, our requirement for an analog will also change as we start to learn more. So yes, I think there's a lot of undertapped resources there too.
Okay, thank you. When you say the analog sites are plenty in India, there is a question from Rakesh Rao. Can you please tell us a bit on the work that you did on runoff catch?
Okay, so basically this is very new work. We collected samples and now they are. So the idea is we're looking at the microbes within the salt crystal. So it's, we're wanting to know what microbes can survive in there, what biosignatures they can look at. And the idea is we were supposed to be coming back to India this year and this summer, and we were going to bring over some of our equipment to collect cores, and we could look at volatile organics and the organic geochemistry, the organic of the molecules as well. But obviously things are on hold at the moment. So there's a couple of projects that one of my postdocs has started to work with. We've been doing work with Amity University and Pune University as well, looking at some of these analog sites. So it's very much looking at microbes in there from a perspective of survivability and biosignatures.
Okay, thank you. There is one question, because you talked about astrobiology beyond science. Maybe I don't know whether we can take it in that way. I'm just pasting it on the screen. This is from Hazarika. What kind of development is done in the area of biosensors for exploration in astrobiology?
So I think this is an area that's people have spent time looking into. So there's been things. There's a couple of kind of biosensors that have been developed, such as the microchip, the life detection chip, which was proposed to go on ExoMars but didn't get selected. There is work going on that, but I think what we find is, particularly with a lot of the like NASA and ESA missions, is the people that kind of develop them, develop the missions are focused on habitability. So looking at the geology of the site, and for that they're using very much instruments that we know and are kind of like the workhorse. I think biosensors are a potential for the future, but I just don't think we're at that way yet to do it. But there is work going on in that area.
Oh, thank you. Do we have a couple of minutes? There are just two more interesting questions.
Yeah.
Okay. Just pasting one more. This is from Viswas. Just a second. Okay. The question is, are there any works going on the analog sites related to the lava tubes of Mars, or the Martian lava tubes are identified to have lesser amounts of solar radiation? Do they have microbes?
So, from my knowledge, I know of a couple of groups that are working on lava tubes. They're very European dominated. So we're looking at the Azores in Portugal. There's some really lovely analog sites over there, and also in Iceland. From my understanding, there are two areas, analog sites, which are being used to look at lava tubes. I don't know of any activity going on in India, but if there is, I'd be happy to hear about it. But yeah, there is definitely work that's kind of, they've identified the fact that there is less amount of solar radiation, and these are places that we should start to consider. So there is some work ongoing at the moment.
Okay, thank you. I have one question. when we talk about sulfur-rich conditions, why don't we look at phosphorus-rich places? Is that important?
Yeah, I think they found the dominant evaporites that they found on the surface of Mars are sulfates, then chlorates and perchlorates. There is some work done relating to perchlorates, and again, Analogue work done in the Atacama has been looking at that. So there are it's not just sulfates. Most of the work to date had been done on chloride. So the fact that there are people now starting to look at sulfates is quite interesting and exciting. So I think, over time, as we start to learn more about Mars and the composition of Mars, we will progress with the Analogue sites and the work that's done in this area.
Okay, good. There's one last question from Karmakar. What is the better method to detect the presence of life? Searching for organic signature or conducting a culture with the samples?
So I think if you're looking for presence of life, for example, on Mars, and you had samples coming back from Mars sample return, I think for organic biosignatures, you're probably still going to go for a GC, so like pyrolysis GC, rather than doing culturing experiments, because it just takes up too much sample and it's destructive. Whereas some of the, if you did Raman analysis, for example, it's a non-destructive method, so you could use the sample for other stuff as well. So I think it's if you had all the sample in the world, yes, you do some culturing as well as that in parallel, but the samples that we're going to get back, for example, from our sample return is going to be so low. So, it's very tiny, not that much, that we need to kind of think about more than just a method. We need to think about a destruction versus non-destructive method as well.
Okay. So one question which I have received here. Can you please explain more on the 0.5 low water activity area, right?
Yeah, so microbes can live in low water activity. So we look at that from a salt perspective. So we can think of your helophiles, particularly, that's focused on that, so high salt. But it's also sugars as well, and kind of so it's kind of like microbes can live. Water activity is really important for life. So when you're looking at these really low water activity, you're kind of pushing the boundary. And what we found is that there's the fungi are actually more resilient to water activity than bacteria in general. So so it's very much dependent on the composition of the fluid and what's in it. So salinity and sugar both have an effect on water activity.
Okay, so there is another question. Just a second. Let's paste it into the screen. Okay, the question is from Kumar. We are doing experiments on the International Space Station. How much accurate are those to that of life on Mars? Hello? Dr. Karen, can you hear us?
Yeah, so the experiments that we do on Mars is we take microbes. I can, yes. Can you hear me?
Yes.
So the experiments that we do on the International Space Station is not like looking. We're not looking at the life on the space station as per se. What we're doing is we're taking microbes up there to learn, and obviously going to the International Space Station is a lot cheaper and easier than going to Mars. So it's kind of a stepping stone to finding out to understanding life on Mars. So it's looking at how microbes would if they would survive, but more importantly, how their biosignatures would be affected by radiation. So this is important when we're looking at biosignatures and the instrumentation and understanding the data that we'd receive from the missions. So it's kind of a stepping stone to find, to understanding life on Mars.
Oh, nice. Thank you. That is a very, very interesting question from Vishmaya. Apart from contamination, how else can life be transferred to Mars from Earth?
So there's work being done on this regarding lithopanspermia. So, for example, for meteorites, we can have life being transferred or stoned. There's work that's been looked. There was a simulation, an ISS low Earth orbit experiment where they called the stone, where they basically put microbes on the outside of a stone and took it into orbit and looked at microbial survival as it entered the Earth's atmosphere. So lithopanspermia is kind of the transfer of life on kind of rocks is one way. If there was an impact and rocks got transferred from one planet to another, that's one way of doing it, which potentially naturally could have happened on early Earth, early Mars time.
Thank you. There's one question which I have received with respect to the places where we can search for life. Apart from Mars, Enceladus, and Europa, is there any other solar system object that you suggest that may harbor life?
I think we're learning more as we go along. There's people talking about some of the other icy moons as potential for habitability. We're also just, for example, talking about Titan. I think as our understanding of these environments develop, I think we will also look for habitability. And I think exoplanets potentially are a source for habitability. There's a lot of work that's been looking at exoplanets from a habitability perspective. So yes, I think we're focused on icy moon and Mars because we know they either do or had liquid water as we know it, and also we can get to Mars in 6 months, we can get to the icy moons within 7. So they're kind of feasible within our lifetime to study this work. But there is work doing on exoplanets at the moment, looking at the atmospheres to deduce if they could potentially be habitable as well.
So do you plan to do any simulation experiments on the moon or samples that are brought back from the moon? There is a question I'm just shortening that.
So the moon,
with the moon samples, are you planning to do any simulation experiments?
We can. We can simulate conditions on the moon. The only work that I've done regarding the moon was looking at more from an in situ utilization thing, where we could use microbes to produce resources if you were going to put a moon base. Produce a moon base. We've not really looked at it from a habitability perspective because it's not really one of those. It's something that we don't really think about as a target for that. But we've looked at it more as an in situ resource project.
Okay, I think we ran more than 1 hour. So there are a few questions, but I think we stop here. We will ask them directly to students. So thanks a lot, Dr. Karen. We have learned there are more exciting questions from the students, a few more coming in. We'll request the students who are watching this to go through the work what Dr. Karen has carried out. So thanks once again to the speaker. Thanks to the organizers and thanks to all the participants. Thank you, Dr. Karen.
Thank you. Bye-bye.